a recorded example of a lesson, primarily the introductory part of the Plants and Transpiration teaching pack. The Plants and Transpiration teaching pack is fantastic because it's very, very short. It can last as little as an hour to an hour and 15 minutes. The experiment, however, may take from 4 up to 24 hours. It should normally, though, be done in around 4 to 6 hours. The curriculum covered within this topic is describing how living things are classified into broad groups according to common observable characteristics, planning different types of scientific inquiries to answer questions, using test results to make predictions to set up further comparative and fair tests, reporting and presenting findings from inquiries, including conclusions, and identifying scientific evidence that has been used to support or refute ideas or arguments. Again, these experiments contained within this are very cheap. The total cost is £6.99, although it can be cheaper if you demonstrate the experiment rather than allowing the class to do it themselves. The resources you'll need to pick up will be food colouring, a white flowering plant, we've trialled it with carnations and chrysanthemums, and plastic cups. So here's an example of the introduction that I've been giving children while testing, to sort of get them up to speed on the science. So the first question I ask is, what is biology? As students study chemistry and physics as well, it can be quite hard to differentiate between the sciences quite young. When we ask this question and ask children to write down their answers, almost every single answer always includes the word life. Although some children are much more anthropocentric and tend to focus on humans. So biology. So I throw these questions out to the class, by the way, and trying to get the whole group involved, is the study of living organisms also their life processes, also involves things that may affect living organisms. It's important also to remember that we're not just studying this now, we're also studying the past, the present, and the future. Okay, after this, since most children tend to focus biology on being alive, we ask the question, well, what is being alive? For the purpose of primary learning, there are 4 life processes. Which are movement, including movement in plants. So although you might not see plants moving around rapidly, they certainly don't have hands and legs. They do move towards the sun and towards nutrition. Also, growth, so the reproduction, which in plants can be sexual or asexual. And finally, taking in nutrition. This is important to remember with taking in nutrition that animals, they eat other things. Whereas plants make their own food by photosynthesis. Now the kids should have a grasp on what it is to be alive. It's important for them to know how to group, how to classify different living things. So, living organisms can be classed into different groups. Some students might throw out vertebrates or invertebrates, but we're not really looking for it on that depth of scale at the moment. So, looking at animals, looking at microorganisms, and we're looking at plants. Now with plants, it's important that the students know there are two types: flowering and non-flowering. And flowering are the focus of the lesson today. So, as we're focusing on flowering plants, I ask the students to name the parts of a plant and the functions of the plant. So, you have at the top a flower, which is for reproduction. You have stem, which is important for movement, as in towards light, and for transport. You have leaves, which are important for nutrition. It's because leaves are the site or predominant site of photosynthesis. Photosynthesis is where the plant uses light plus carbon dioxide plus water produces food or carbohydrates and oxygen. Lastly, you have the roots. So the roots take in water and minerals. Also, they produce another function. They anchor the plant. What I like to do to explain how anchoring works is I get a student, take them up and help them round. Then I ask two other students to come and hold the other student and be the roots. Then I try and move the students around. And I say they can't be moved because they're anchored. But now we've got a problem. We've shown here in the leaves that we need water. But the roots are the main site for taking up water. So we need some sort of transport system for the water to get to everywhere else in the plant where it's needed. It does this by a process called transpiration. Transpiration works by water constantly evaporating through tiny holes in the leaves. These holes are called stomata. And it works a bit like sucking up a drink through a straw. This evaporating through the holes creates a force that pulls water from the roots up the stem to the flower and to the leaves. This is called transpiration. And it's going to be the focus of our experimental lesson today. So the experiment uses food colouring, and you cut a plant and place it in the water and food colouring, and it should, after time, change the colour of these. So if we put green food colouring in with all the water, it should get taken up by transpiration through the stem to the flower, hence changing the colour. With the experiment, it's important when we're cutting it to cut it below water. Otherwise you can get air bubbles in here which stop transpiration. The way I like to think about it is getting a straw and then pinching it, and that's the way I demonstrate it to the kids in the class. By pinching it, that's like an air bubble which stops me sucking up more liquid. So the video, there's already a video demonstrating the experiment, which you'll be able to see my hands and see how it goes forward. But also it's important to state that there's worksheets for the children to complete as part of the teaching pack, if you require them to do so. Finally, I'd say there's a thing they can do with the extension work, which is after their experiment has completed, whether the flower's changed colour or not, what they can do is they can cut down vertically down the stems, and then they should be able to see whether the experiment was successful or unsuccessful food colouring that has highlighted these tubes that exist within the stem and dyed them. And if there's been an air bubble or a problem, you should be able to see it dyed up until that point.